Hello, my name is Francis Kilkenny. I'm with the U.S. Forest Service Rocky Mountain Research Station in Boise, Idaho. I am going to be talking about really what's local. Really taking a methodological approach and a conceptual approach to measuring patterns of local adaptation. I want to go ahead and thank some data co-authors for some data I'll be presenting at the end. Holly Prendeville, Brad Sinclair, Rob Mazzotti, Bryce Richardson, Steve Larson, and Blair Waldron. None of this would be possible without collaboration and support from Fred Edwards, Peggy Olwell, Sarah Kulpa, John Proctor, and many others. There are postdocs and technicians in my lab that have helped with the projects that have gone into this thinking. Sarah Barget, Jeff Ott, Jessica Irwin, Kimberly Stocks, Michael Ottenlips, and many others. So I also want to thank some of the funding where we've gotten funding and support. Great Basin Native Plant Project, which I am the co-lead with Fred Edwards with Forest Service, BLM, and Fish and Wildlife Service. So thank you very much. So when we think about local adaptation, especially in the context of restoration, I think one way to think about this is to just from take these quotes from a paper that I worked on with some others internationally. Choosing the geographic origin, the provenance of seed use and restoration is an early and fundamental decision faced by restoration practitioners. The traditional practice has been to source seed locally to the restoration site. That's called local provenancing, because it is expected to maintain the evolutionary history of plant populations, minimize maladaptation, and limit deleterious genetic effects. So in this case, we defined local adaptation as superior fitness of a local population over other provenances when grown in its home environment. And a local provenance is defined as seed from either within the restoration site, which is called strict local provenancing, or within close geographic proximity to the restoration site, which is relaxed local provenancing. Defining a local provenance, however, is a difficult task and is rarely articulated well. Adopting a genetically informed definition of local provenance is the most useful approach. However, such an approach is perhaps the least attainable for practitioners because of the costs, time, and skills required. So one of the ways we define provenances and try to deal with local adaptation in restoration practice is to use seed transfer zones. And seed transfer zones allow us to depart from strict local provenancing. So in this case, departing from strict local provenancing strategy relies on the development of robust seed transfer guidelines, such as seed zones. Seed zones refer to regions that have similar environments, and transfer within zones should result in few detrimental effects on mean population fitness. So what I want to do is give you a conceptual taxonomy of seed transfer zones. So as you can see here, this is my taxonomy, and I will go through it kind of in general and then more specific detail. There are a lot of different ways seed transfer zones can be developed, and I think it's really important to have an understanding of how that occurs so you can understand what to use when and what type of information might be more valuable than other types. So one of the largest divisions that, best well-known, I guess, the most well-known division is between provisional and empirical seed zones. So provisional seed zones, the most well-known are the generalized provisional seed zones of Bauer et al. And I'll talk about that a little bit later. But you can also have provisional seed zones that are species specific. And then there are empirical seed zones, and the key definition here is that there is genetic or adaptive information that is being used, where these plants have been studied in particular, and that thus makes it empirical. Another division is generalized versus species specific. So again, the Bauer et al. Are generalized provision or generalized seed zones, and then empirical seed zones, or sorry, species-specific seed zones can include provisional seed zones, as well as empirical. And then lastly, within the empirical seed zones, data is either from common gardens, which is quantitative genetics, or population genetics from DNA markers. So the traditional seed transfer zone development really comes from trying to measure local adaptation in some way. I'm really glad that Owen gave his talk earlier, because some of the definitions of the signatures will help in understanding this. So the best way to develop seed transfer zones in terms of understanding local adaptation is to use a reciprocal transplant study. These are very accurate on a species basis. It measures that signature 3 of adaptive variation, which is really a direct test of local adaptation. And it has the highest standard of comparison. It's that gold standard. And what reciprocal transplant studies do is they take all the locations where source populations are and then move the source populations between those locations. So you get a complete picture of how transfer can happen. However, reciprocal transplant studies are extremely costly, especially if we're doing them on the scale that we need to do them for seed transfer zone development. Often reciprocal transplant studies are done on a very small scale. This is a case here where Wang et al. Worked with lodgepole pine in British Columbia. And this was a massive program that happened over many years. And it had, I think, over 60 sites and roughly the same amount of collections. So it's very hard to do these, but it does give us the best data. So an alternative that has been used quite often are gene-ecological common gardens. This is measuring signature 2 of adaptive variation, which is the trait by environment, where you look at the source environment and you compare it to the traits that you find in the common garden. The common gardens are usually one or a few common gardens. It's accurate, relatively accurate on a species basis. It has well-documented success, especially in forestry. It doesn't test directly for local adaptation. So it's really testing that signature 2, which is a signature of local adaptation, but it's not a direct test. And they are still relatively costly, but they're effective. So because of these costs, a lot of people have decided to look into alternative methods for seed transfer zone development, mostly because they're more cost effective. But the question is really, do they work and what do they tell us? So probably the one that's been the most used are the generalized provisional seed zones that Bauer et al. Put out. People have also used ecoregions from Amernick et al. 1987. One of the huge advantages of these generalized seed zones, and the answer is to the question for the generalized seed zones, minimum winter temperature and annual heat moisture index were used to develop those seed zones. And then they were meshed with the ecoregions. The advantages of this is that they're very inexpensive. It's just a computer exercise, GIS exercise. They're generally applicable. So even species that don't have data, empirical data, you can use these seed zones. However, they're not really very accurate on a species basis. So they're useful until you have better data. Ecological niche modeling gets into a little more specifics. So it's a species specific. It uses herbarium specimens for the most part, and then tries to model what seed zones might be given the relationship between the locations of the herbarium specimens and the climate at those locations. It's inexpensive. However, we don't really know how accurate it is on a species basis. It could be quite accurate, but we're not really sure. And we really need to test the predictions of these models. And so far, they have not been well tested. So they've definitely been proposed. Several papers have been put out that have these species-specific provisional seed zones, but we don't really know how effective they'll be. Another way, it's kind of a different approach, is to use population genetic structure to understand how lineages are distributed within a species. This is relatively inexpensive, especially nowadays where we have cheaper and cheaper access to DNA technologies. It's accurate on a species basis. It addresses genetic issues like outbreeding, depression, and genetic variation, but it doesn't really address adaptive variation. So folks have also tried to use these DNA markers to predict potential adaptive variation. Again, it's relatively inexpensive. It's perhaps accurate on a species basis, but does it address adaptive variation? Well, we're not really sure. What they're using are these what are called outlier loci, which allows, when you look at those loci and you correlate them with climate, you can find out maybe whether there's loci that are showing local adaptation. However, again, the predictions of these models really need to be tested, and they haven't really been tested in any thorough manner. So we don't know how good these are. So how can we test these alternative methods? My proposal is to really use this common garden array approach, and I'll give an example of that after this. It compares favorably to reciprocal transplant studies. Essentially what you're doing is you're taking more common gardens that you would use in a gene ecological approach, but not quite fully reciprocal transplant. And then taking as many populations as you can and planting them across that array. It can be used to test alternative methods. It is less costly than reciprocal transplants, but more costly than gene ecological studies. And we can link it to other data. So we can, it's easy to make ecological niche models. And if you also take genetic data along the way, we can link it to that. And through this method, we'll be able to test those other alternative methods. Essentially what we end up measuring in a common garden array is this transfer distance. So this is a difference between the environment at the garden and the environment at the home site of the sources. And so we can map these onto this transfer distance. So in the center there, that's what you would see at local gardens, or where a local source is at a local garden. There are multiple gardens here and they're all overlaid with each other. So it's kind of a conceptual, it's an abstract concept here. But essentially what we're measuring is the scale of local adaptation. One of the cool things about this is that the number of test sites you use rapidly increases the precision of your estimates of the scale of local adaptation. So the more sites you have, the better, but there are diminishing returns after about 12 or 15 sites. So I want to lastly just kind of go in quickly through a common garden array in practice with blue bunch wheatgrass, and then I will end the talk. So first, I just want to point out that with our blue bunch wheatgrass study, we did do the genetics. We still haven't done the comparisons to determine how good genetic prediction might be. But a quick thing to point out is that we covered quite a bit of adaptive variation or genetic variation in the study. And the genetic variation that's actually in most of the cultivars is quite a bit smaller. So just something to be aware of. Essentially, there are 4 different sections of genetic structure that were put in the study. We had 15 common gardens in total. We had a set of northern common gardens and a set of southern common gardens. These are arrayed across Oregon, Washington, Nevada, and Idaho. On the right-hand side here, these are this is the environmental space that's being looked at, and the standard deviations around the common gardens. So you can see that 2 standard deviations around the common gardens in environmental space. So you can see the gardens do a pretty good job of covering the environmental space of the populations that are being put into these gardens. All the smaller points are the populations. So this is what we get. This is these transfer distance functions. And I don't know that I want to cover this slide that much, just to say that we had some significant results, particularly in the northern common gardens with all three fitness measures: size, reproduction, and survival. We got somewhat of a good result with reproduction in the southern gardens. But what we can use for this is to actually measure how much difference in a fitness trait we would expect given a certain distance of transfer. And this is environmental distance, and this is in standard deviations, so it's kind of abstract, but this can be mapped onto the landscape. So as you can see in all of these, when you're moving away from local environments, at least in one direction, in some cases, and both in others, there will be a loss of fitness at a certain rate. So ultimately, this is how I believe we can measure local adaptation, and then linking this into some of those other genetic methods, we can determine whether those alternative methods work well. So one very last thing I want to show you is the costs of a typical common garden study on a per species per garden basis is about $120,000. The blue bunch wheatgrass per garden basis was around 80. And then the study that Sarah is going to talk about was down near 60. And with the blue bunch wheatgrass common garden, if you were to just do that consecutively, it would take quite a long time to get through a whole bunch of species. But what we're doing with the Great Basin Forb study we're doing multiple species at the same time, and so we're reducing that per species time to completion by a whole lot. So thank you very much.